In this video, I'm going to show you how you can import the measurement we exported from QPath into Cytomap and then visualize some measurement within figures in Cytomap. So first of all, we need to make sure that you have the correct Cytomap version. So if you type in Cytomap download, for example, then you're going to find the GitLab of the kernel lab that developed Cytomap. If you go on this GitLab, so then you'll see that Cytomap is based on MATLAB, but you don't need MATLAB to run it. There's standalone versions. Unfortunately, you need to have Windows because those standalone versions they only work with Windows. If you don't have Windows, you'll need to have MATLAB, and then you can use Cytomap. If you have Windows, you can use a standalone installer, so you don't need MATLAB. Just make sure that you take the correct version. So the latest version is 1.4.21, so you can go down and find the correct executable file. When you download this, then you can run the executable file and it will install it on your computer. It's already installed on mine, so I don't need to install it. So to download it again, I'm gonna open, so if I type in Cytomap in Windows, so you can't see because it's on my other screen, but the Cytomap icon opens, and then Cytomap window, the main window, Cytomap window, opens like this. So now I can open the measurements. So if I go into File, if I have multiple samples, so let's say you have multiple images coming from multiple samples, either biological replicates or different conditions, you can load multiple samples. Just if you do that, just make sure that you then select all the files before opening them. Otherwise, at least I run into problems. Here we only have one file. So you go to load table of cells. I'm going to go back to my QPath project, and in this I'm going to select the measurement file. And then, as I told you, Cytomap works with point clouds. So the first, so here there's a window that pops up loading the data, and then Cytomap will ask me where the coordinates of my different objects are, asking what corresponds to the x-axis. So here it's going to be the x coordinates of a centroid. Then the y-axis, which is going to be the y-coordinate of the centroids. And then it also asks for the z-axis, because you can also work with three-dimensional data. Here it's not the case. And in that case, you can tell Cytomap that there is no z. That's the default option. And then Cytomap will know that I only have x and y and not z. And then you have window that pops up that summarizes all the information you have. So here tells me that I have the image information, x and y coordinates, then information for FOXP3, CD4, and CD8, and the fake z coordinates. So I can load the data. And then I can look at my data, so I can create a new figure. So when you create a new figure, you have a window that pops up like this. And here, so every single dot corresponds to a cell. And so here you can actually keep the x and y coordinates and select a measurement for the channels. For example, I'm going to select FOXP3. And now I have the expression of FOXP3. If you look at this, you can recognize the tissue. You just see that it's upside down. It's because in MATLAB, X goes from left to right, as in QPath, but Y goes from bottom to top, while in QPath it goes from top to bottom. So if I want to visualize it in the correct way, I can click on Options. I have another window that pops up, and then in the y-axis I can invert it, and now I can see my tissue again. And if I want, I can change the range of intensity. For example, I can put the maximum of intensity to 10,000, for example. And then if I go back to QPath, so here I only have FoxP3, and it seems that it makes sense. I see that I have a higher intensity in the tumor, especially in the tumor periphery and in this region, and that's what I see here, so that seems to make sense. Except that now I only have, dots for each cell. I don't have a fluorescence intensity over my image. I can also visualize other measurements. So for example, I can look at the CD4 in x-axis, CD8 for y-axis. So now I'm going to invert back. Well, it doesn't make sense. I can look at the FoxP3 intensity in those cells, or I can look at the density, for example, point density. And now it very much looks like what you would have when you do cytometry, and you actually have tools. For example, here you can define windows, and so then you can do some gating to define positive cells for CD4, for example, and CD8. Or if you were to have double positive markers, you could also define a gate for the double positive markers.